The human nervous system and the digestive tract are not distinct entities. They operate in absolute tandem, forming a single, continuous biological network. For decades, psychiatry viewed conditions like severe depression, anxiety, and cognitive impairment through the lens of isolated neurotransmitter deficits. Clinical data now suggests these symptoms occur as downstream effects of systemic immunometabolic dysregulation. The primary vector for this inflammation originates in the gastrointestinal tract, driven by modern dietary mismatches. This connection is the gut-brain axis. As shown in this anatomical model, a two-way vascular highway connects the gut mucosa directly to the brain, allowing signals to travel constantly between the two regions. To treat severe cognitive and psychiatric symptoms, practitioners must trace the pathology back to its origin by analyzing the cellular mechanics operating inside the gut lumen. The intestinal barrier holds back a massive, highly active microbial load. Yet, this protective wall is remarkably thin, consisting of a layer just a single cell thick. Between these cells are biological gates known as tight junctions. In a healthy gut lining, these junctions lock the individual cells together, shielded beneath a thick, protective layer of mucus. This system evaluates particles, allowing essential nutrients to pass into the bloodstream while blocking pathogens. This sorting mechanism is vulnerable to ultra-processed foods. The most damaging components are synthetic emulsifiers, like polysorbate 80 and carboxymethylcellulose. The human body does not fully digest these synthetic emulsifiers. Instead, they act like detergents. As they pass through the digestive tract, they dissolve and erode the thick mucous membrane lining the gut. With the mucus layer stripped away, the fragile epithelial cells are left exposed to structural damage and direct bacterial invasion. Simultaneously, high concentrations of saturated fatty acids in these foods alter the composition of the microbial community itself. This shift is called dysbiosis. As beneficial bacteria decline, the gut loses the essential byproducts those microbes normally produce. The most critical missing byproducts are short-chain fatty acids, specifically butyrate, which the body requires to maintain the structural strength of the gut barrier. Emulsifiers erode the external shield, while saturated fatty acids cut off the supply of butyrate. This combination strips the gut of its biological armor, priming the system for an inflammatory breach. That breach is known as increased intestinal permeability, or leaky gut, occurring when tight junctions structurally give way under constant chemical stress. Structural failure is exploited by lipopolysaccharides, or LPS. As junctions pull apart, LPS molecules slip into the bloodstream. The systemic immune system detects this invasion and mounts a defense. Immune cells flood the bloodstream with inflammatory markers, specifically C-reactive protein, interleukin-6, and TNF-alpha. This saturation of inflammatory markers produces a specific clinical state known as cytokine sickness. We can observe the effects of this state in clinical trials. Healthy human subjects injected with inflammatory LPS proteins report acute depressive symptoms, irritability, and fatigue within 20 minutes. These sudden mood and cognitive shifts occur as direct, measurable biological reactions to circulating bacterial toxins. When a patient presents with severe brain fog or acute depression, their brain is often functioning exactly as it should, interpreting and reflecting a gut-derived systemic alarm. Once in the bloodstream, these inflammatory cytokines and LPS molecules travel toward the boundaries of the central nervous system. The blood-brain barrier protects the brain, but chronic systemic inflammation eventually compromises its physical integrity. Under the assault of these inflammatory particles, the biological mesh begins to fray and separate. With the barrier degraded, neurotoxic elements, specifically LPS and circulating saturated fatty acids, gain entry into the brain tissue. Waiting inside the brain are microglial cells, the resident immune defenders of the central nervous system. As LPS and saturated fatty acids encounter a microglia, they lock into the TLR4 receptor. This triggers a chain reaction, activating the NF-κB pathway. The cell then transforms into an aggressive attacker, releasing its own inflammatory cytokines into the brain tissue. This localized neuroinflammation damages surrounding neurons. As the neural tissue degrades, cognitive function, focus, and memory become impaired. Chronic microglial activation provides the physical substrate for many neurodegenerative diseases and treatment-resistant mood disorders. This confirms that dietary inputs can rewrite the immune posture of the brain. Understanding these mechanisms forces a shift toward a systems biology perspective, guided by evolutionary mismatch. Standard pharmacology often fails to treat isolated psychiatric symptoms, ignoring modern environmental stressors. This model outlines these overlapping dysfunctions. Tracking upstream variables like microbiome disruption and circadian misalignment reveals how they converge to produce neuroendocrine dysregulation and clinical disease. Recovery requires a framework that targets these upstream regulatory processes. This table maps out the specific interventions and physiological targets required to restore metabolic flexibility. Those interventions include realigning circadian rhythms, eliminating synthetic ultra-processed foods, and utilizing targeted supplementation, like L-glutamine and zinc, to rebuild damaged mucosal tight junctions. Mental and physical health share the same mechanistic pathways. Protecting the integrity of the gut barrier is the direct requirement for protecting the mind.